Les mécanismes de la douleur sont-ils les mêmes chez les mâles et les femelles? Les expériences réalisées sur des souris par l'équipe de Jeffrey Mâles montrent que ce n'est pas le cas. Mais elles prouvent surtout qu'on a tort de penser qu'en recherche, les résultats obtenus en étudiant des animaux mâles sont aussi valables pour des femelles. Une révolution.
For the past 15 or 20 years, one of the major new developments in pain research is the idea that neurons are not the only cells that are processing pain in the nervous system. We've come to learn that cells of the immune system, glial cells, are also very important as well. And this, of course, was a major new realization because it used to be thought that these cells were essentially glue doing nothing but filling in space and supporting the neurons. The best evidence has come for the role of a cell called microglia, especially in the spinal cord. Microglia are now known over hundreds of published experiments to release a number of factors that then communicate with neurons so that pain signals can be sent up to the brain from the spinal cord. What we found, briefly, is that this is very true that microglia are very important for pain processing, but only in male mice. If you do the very same experiments that have been done with male mice and rats over the years, and you do those same experiments in female mice, what we found was that microglia, simply put, have nothing to do with it. You can block microglia in various places, you can even kill them off. This has the expected result on pain in male mice, that is, it blocks it. But in female mice, we found out that these procedures had absolutely no effect whatsoever. It's important to note that the amount of pain that was being displayed by male and female mice was exactly the same. So we inferred from that pain processing in female mice, if it doesn't use microglia, must use another type of cell. And indeed, we found some evidence that the cell in question are T cells of the adaptive immune system. Why wasn't this noticed before? Well, virtually all the research that's been done in this field, and indeed almost all the research in pain, and almost all the research in biomedicine is performed on male rats and male mice. It seems to me that the days that we can make the assumption that it is okay to use male animals to study a syndrome that affects men and women, I think those days are numbered, and our findings should really serve as Exhibit A as to why preclinical research from the very, very early levels really needs to use both male and female animals.